Okay, hi Rasid, how are you?
I'm fine, sir. What about you?
I'm also fine. So right now feeling comfortable,
Yeah,
ready for interview.
yes, sir. Sure.
So we can start.
Yes.
Okay, so Rasid, just give your brief introductions about yourself, so we can further start.
Okay. Uh, Good evening, sir. Myself, Raisuddin Khan. I am actually from Aligarh, Uttar Pradesh, but I am born and brought up here in Arunachal Pradesh. I have done my schooling there from itself. For my graduation, I came to Assam. I joined Dibrugarh University for my bachelor's in botany. And for my PG, I have joined Aligarh Muslim University in the course MSc Microbiology. After getting my PG in 2018, after that, I'm solely preparing for government services.
Okay, so your masters in microbiology, right?
Yes,
Okay, so I think your area and topic will be microbiology or immunology, something like that.
yes, sir.
Okay, Rashid, tell me, for the bacterial identification, there is a process, right? There is a process known as molecular identifications. So, for the identification of a new strain of a bacteria or new species of bacteria, what is the procedure for that? How researchers or research scholars can identify or can name a new species or strain of the bacteria?
Sir, the molecular detection includes the most preferable method is doing the 16S rRNA sequencing, which is the most probable and which gives the most accurate result till which is very much prevalent in the present time. It actually derives its name from the 16S subunit of which is the small subunit of 23S rRNA.
So why only 16S rDNA is considered for identification of a bacterial species? Is there any specific reason behind it?
I'm sorry, sir. I mistakenly said that it is 16S rRNA. It is 16S
rDNA,
rDNA.
yeah.
Sir, basically it have some conserved sequences which are very much which are very much conserved, and they can be used for identification up to the specific species level.
Okay, Rasid, when we use conserve, that means conserve sequence, that means there is no chance of mutations and there is no chance of any alterations in that sequence of a DNA. Okay,
Yes.
so if a sequence is conserved, then how it is different in different type of species of bacteria?
Sir, basically, these bacterial species differ from each other on the basis of certain functions. They have certain peculiar characteristics. These are governed by certain genes which are known as housekeeping genes. They are actually present in this conserved sequences with the this information which we exploit in the identification of the species.
Okay, Rasis, suppose if you have two bacteria, right, and you extracted the 16S rDNA from these two bacteria and sequence them, are there will be differences in both sequences of the DNA, bacterial DNA, or they will be the same because they are the we are dealing with the 16S rDNA, so they are 16S rDNA are the conjugate sequences. So will we find any differences in both DNA sequences or the same?
Sir, there will be differences, but that differences will be of very minimal amount. As these are the
So
bacteria only.
what kind of differences will we observe in that both DNA sequences?
I'm sorry, sir, I'm not able to understand the question.
Okay, okay. So that means my question was that how the two bacterial strain 16S rDNA are differ from each other. Is there any structural differences? Is there any number of gene sequence, number of DNA sequences, etc.?
Sir, in the difference lies in the order of the nucleotides
Very
in
good.
the
Order.
stretch of that conserved.
order of
order, yes, sir.
nucleotide. Okay,
Yes, sir.
that means if we examine the two bacterial rDNA, 16S rDNA, then we will find the order of a nucleotide base pair sequences, that is ATGC. There will be a difference in the ATGC sequences, right?
Sequences, yes, sir.
Okay, got it. Uh, Rasil, for clear identification of a bacterial DNA, bacterial species, is there any criteria of number of a nucleotide? Of what should be the minimum number of nucleotide will be there in DNA sequencing for considering molecular identification of that DNA? Is there any number of a nucleotide needed? Suppose 100,
I'm
200,
not so
300,
sure
400,
about it.
500.
Sir, I'm not so sure about it, but I can guess. I think it is less, more or less than 1,000 nucleotide.
Okay, it's a nearly 1500 base pair.
Okay, 1,500.
Okay,
Yes, sir.
Rashid, for identification of a bacterial species on the basis of molecular, we also need bioinformatics tools. So, which kind of a bioinformatics tools we require for the similarities, to check out the similarities of existing bacterial species for the unknown bacterial species?
Sir, we actually take the help of NCBI, that is National Center for Biotechnology Information,
Okay.
which have a lot of programs. For nucleotide, we have others. For protein, we have separate databases and search engine where we can put our known sequences and we can check for similarity and dissimilarity. Uh,
Okay, Rashid, have you heard about the PGPR, plant growth
yes, sir.
promoting rhizobacteria?
Yes, sir.
Have you heard about it?
Yes,
What is PGPR?
sir. The plant which have its roots in the soil, they the root around the root, it creates a microenvironment, and inside that microenvironment a lot of uh, interaction with the bacteria and the other root bearing microorganisms the plant used to interplay. For example, when the most famous example of the PGPR, we have the rhizobium, which we use for nitrogen fixation. Beside this, we have a lot of other bacteria for phosphate solubilization bacteria are there. They plant secrete certain enzymes, a certain signaling molecule, which actually attracts this microorganism near the periphery of that root, so that they can colonize, they can create a biofilm around this the root node, they on the root, over the root, so that they can interact and they can help the plant in order to degrade the complex substrate which the soil harbors. And after the activity of this microorganism, the plant is able to degrade this the complex substrate, and it derives the nutrition so as to use it for its growth and reproduction.
Okay, okay. Rashid, nowadays we all researcher community as well as farmers as well as agriculture practices, we all are dealing with the chemical fertilizers, and as we all know, chemical fertilizers have their negative impact on the, not only on the soil health but also on the microbial community, as well as on the also on the human beings. Okay, so is there any
So
alternate approach of a chemical fertilizer?
yes, sir, definitely we have. The era which we are living is the era of the sustainable development,
Yeah.
and not only sustainable development in the in broader perspective, but in terms of agriculture, we are using biofertilizers. We are using the the help of biotechnology to create new genetically modified microorganism which we can use for uh, uh, increasing the productivity of the soil and the crop by itself. For example, the best example is the biofertilizer, where we are using the carrier-based microbial suspension, which we use to amalgate in the soil so as to get alternative of this uh, chemical fertilizer which have very negative um, consequences, and they harm the environment in a negative way.
Okay, in your one answer you used term phosphate solubilizing bacteria, that is PSB.
Yes,
What
sir.
is this, and what is the role of PSB for the growth and development of the plant?
So, sir, phosphate, which we have in the soil, it is very complex in nature, and it is not, although it is present in the soil, but it is not available for the plant to absorb and to get assimilated inside the plant. So there are certain group of microorganisms which we count in the broader category of PSB, that is phosphate solubilizing bacteria. They uh, they they they, they digest this complex form of phosphate to in order to make it in convert to in the ionic form of phosphate, that is PO4, which can be assimilated by the plant, and it can be used by the plant for its growth and development.
Okay, very good. Well done, Rashid. When farmers and we also applied chemical fertilizer, that is DAP, diammonium phosphate, as a source of a phosphate element in the chemical fertilizer form to the soil, what will happen? How a phosphate will form a complex, and what are the complex that form in the soil?
So, so when this chemical fertilizers, like you have mentioned, that DAP, that is diammonium phosphate,
Yeah.
it will uh, it will produce it will give only two elements to the plant, that is nitrogen and your phosphate. That phosphate, when it comes with the contact with the moisture or water, it will form phosphoric acid H3PO4 or H3PO3, like this. But when this phosphate solubilizing bacteria come in contact with this complex nutrients, they degrade, in a way they hydrolyze the H3PO3 and H3PO4, so that the phosphate form is readily available to the plant. For that, after that, the plant can easily assimilate all this phosphatic form.
Good. Well done, Rashid. As we know, in the soil, phosphates are present in the both form, that is organic as well as inorganic. Which
Sir,
form of a phosphate can be utilized by or can be uptake by the plant?
Sir, basically, soil is a very dynamic environment. It harbors both complex and uh, the degraded form of every micronutrients as well as macronutrients. As far as the phosphate is concerned, the inorganic form, that ionic form, is the requirement of the plant. As a result of which, if it is present in the ionic form, it can be uh, get inside the plant body in the form of ion, with taking the help of water.
Very good. Rashid, for transportation of any kind of a molecules from outside to the inside of the cells or inside to the outside of the cells, there are some mechanism, there are some processes like as a diffusion, osmosis, transportation, etc. So for transportation or for uptake of a phosphate from the soil to the plant cells, there are a is there are any specific molecules for their transportations, or it is common way to transport from soil to the plant cells?
So definitely we have, for we have diffusion also, we have osmosis also, and there is the involvement of some sodium potassium pump also. For example, if for the plant used to respire, they take oxygen from the environment. It is a simple process of diffusion. By the help of stomata, it can directly enter into the plant microenvironment. But as far as the ions are concerned, they can enter inside the plant body by the process of assimilation by the roots, by the help of osmosis as well as the sodium potassium pump.
Okay, tell me one thing on the logical point of view, Rasdeep. Why bacteria help plants? Why they unnecessarily solubilize the phosphate? What were the form of the phosphate they present in the soil? They utilize their own mechanism to solubilize the phosphate for available to make available for the plants. Why bacteria do these things like that?
Mm. Sir, there is a beautiful quote that there are no free lunches in the Washington DC. That is, nothing is for free. So the bacteria are also selfish in their own in their play. They are helping the plant because the plant is helping them, because the plant is giving them an environment where they can cohabitly exist, and they can the plant is giving them a proper environment where this microorganism in turn can play and reside in a very luxurious manner.
Well done, Rasid. Suppose you are dealing with some microbes and want to examine the surface structure of that bacteria. So, which kind of electron microscopy will you prefer for examination of a surface structure of that bacteria?
Sir, if it is not that much peculiar, we can directly go for the optical microscopy. But if we want to examine the outer structure of any bacteria, there is a electron microscopy, that is the scanning electron microscopy for the visualization of the outer structure of any microorganism.
Rasid, have you heard about the intrinsic protein?
Yes, sir. Intrinsic protein are present in the cell membrane of any living component,
Yeah.
whether it's a plant, animal, or bacteria.
Okay, so what is the role of intrinsic protein in the cell membrane?
Sir, intrinsic protein basically they spans the whole width of the cell membrane. They sometimes act as a channel for the transportation of certain ions and for and sometimes glucose also.
Rasid, hoping that you have heard about the fluid mosaic model of the plasma membrane. What is the meaning of
Yes,
a
sir. I
fluid
have.
mosaic?
Sir, this concept was given by J.D. Robertson. Uh, if I'm not so sure about it, I guess it is given by J.D. Robertson only. It
Singer and Nicholson.
it I'm sorry, sir. Singer and Nicholson have given this fluid mosaic model
Yeah.
of plasma membrane. Basically, it says that the plasma membrane is having this the lipid molecule that is hydrophobic tail inside, which are hydrophobic, and the hydrophilic tail which spans outside the membrane in the both side. They exist in the bilayer form, and they are in continuous manner. They used to exhibit a flowing manner. They are moving inside this the in that microenvironment with the continuous interaction with the membrane, this protein that is the intrinsic as well as extrinsic protein. When we visualize this the plasma membrane inside the microscope, we find a mosaic pattern, mosaic of the this extrinsic and intrinsic protein which are embedded in the whole the plasma membrane of the bilayer membrane. So it gives a mosaic like pattern when we visualize under the microscope.
Good. Rasid, suppose we are working in a laboratory and we want to analyze and examine the location or movement of a plasma membrane protein, right? Membrane protein. We want to examine the movement of a membrane protein. Which kind of a technique will we use for the examination of that lateral movement or peripheral movement of a protein?
Sir, are you asking about the method?
Measuring of a movement of a membrane protein.
Measurement of the movement
Easy to
protein.
way, that is lateral movement of the protein, lateral diffusion of the protein.
Sir, we can you take the help of certain carrier molecule if they are transferring or not. On the basis of that, I think we can measure the
Rasid, have you heard about the
transportation.
FRAP technique? FRAP.
Sir, I have heard, but I don't know what is it all about.
Okay, no problem. Okay, okay. Suppose we are also working in a laboratory and we want to analyze the protein interaction in a cell. Okay,
sir.
so can you suggest some technique which can be used to analyze protein-protein interactions in the cell?
Mm,
Okay,
I'm sorry, sir. I'm not able to recall it right now.
okay, no issue. Suppose the question is same, but the interaction is not between the protein-protein, it's a protein and a DNA. Then what will be the answer?
Interaction of protein in DNA.
Yeah.
Uh, Sir, inside the nucleus, some histone proteins are there which used to which help in the packaging of the DNA, the whole stretch of DNA in a very they forms so that a compact structure inside the nucleus so that the whole DNA can be packed in a very smaller portion of that nucleus. So I think you are talking about that interaction of stones and
Okay,
DNA.
got it. Rasid, have you heard about the immune tolerance term?
Immune tolerance, yes, sir.
So, what is immune tolerance?
Sir, our body have a tendency to maintain a homeostasis. That is, the there is no any disturbance in the internal environment of the body. When there is a involvement or there is a pathogenic attack, our body used to defend it from both the measures by adaptive as well as the innate immunity we have. But sometimes, due to our own cells, that is our own cells, if they are infected with any virus kind of thing or any microorganism or pathogen attack is there, there is a expression of this the immune cells inside the body. At that time, there is a special cell that are known as regulatory cells. They are involved in the suppression of the hyper immunity response, which actually helps the body in order to minimize that hypersensitivity or the over expression of our immune cells. That the whole process is known as the autoimmunity.
So, immune tolerance is beneficial or harmful for individuals? That is, what is the role of immune tolerance? Is it harmful or beneficial?
Sir, it is both harmful and beneficial. Beneficial in the case when there is a overexpression of our own immune cells, it will negatively affect our own organ and the cells we have in our body. But in the patients where they have undergone an organ transplantation or something where they have their own immune system is compromised and they are taking the help of immunosuppressive medicine so as to counter the hypersensitivity reaction, there it can play in a very negative role. They can result in the graft rejection or it can affect in a very negative way by exhibiting the hypersensitivity.
Okay, Rasi, suppose you are dealing with the antibody M
So,
and go for the SDS phase electrophoresis and analyze the band pattern on the gel. So, what will be the number of a band on the gel if you are dealing antibody M?
sorry, sir, I don't know.
Have you ever draw the structure of a antibody M?
So as far I can recall, it is pentameric.
Yeah, it's a
So,
pentamer. Definitely, you are right.
sir, when there is any antigen or immunogen, it the whole the antibody they actually spans in a way that it forms a pentameric structure around the uh, antigen so that they can
Yeah, got it,
they
got
can
it.
kill and
But
neutralize that
my
antigen.
question is that suppose leave it. Suppose you are dealing with antibody G, okay, simplest form of the
Mhm.
antibody, and then what will be the number of a band pattern will be on there will on the gel band?
Sir, two, I guess.
Two. How?
Sir, I guess IgG is bivalent to
IgG is monovalent, not bivalent.
monovalent. Okay, sir. Sorry, sir.
IgA is bivalent.
I got confused, sir.
Okay. How many number of polypeptide chains are involved in the antibody?
Sir, there are actually four.
Okay.
Two are yes, sir. Two are s- bigger and two are shorter. They arrange in a Y-shaped manner, and they have disulfide linkage between this the structure.
Rasid, what different kinds of level of protein structures are involved in formation of a complete antibody? That means functional antibody. For a formation of a functional antibody, what different level of protein structures are involved in that?
Mm. Sir, are you talking about the protein modification from primary to secondary, tertiary?
Yeah.
It, how it moves. So, for example, the primary form of the protein is just a smaller stretch of the amino acid which are linked with the amide linkages. When it enters the secondary modification, some hydrogen bond involved get involved in it. When it moves to tertiary structure, other hydrophobic interaction which we have like disulfide linkage and these things get involved. And when it comes to quaternary structure, it's simple. We can measure by taking if two tertiary structure are coming in contact to each other by having some disulfide linkage or other hydrophobic interaction, it will convert in the quaternary structure. So the antibody, as far as I said, they are multipeptides. They are. It means it have to be in the quaternary structure. It is the last form of protein modification.
Rasid, suppose you have a protein that is circular protein, and the number of amino acids in that circular protein is 120. Then how many number of a peptide bond will be there in that protein?
So 119.
You are dealing with a circular protein, not a linear.
Okay. Sure, it will be 120 only.
121. And
No, sir, 120.
if I ask the same questions for the linear protein, then what will be the answer?
So linear protein will have one minus the number of amino acid.
Very good. And for circular,
Yes, sir. Same number of the number of the bonds will be the same number of the amino acid. 120 amino acids means 120 bonds.
okay. As we all know, that is there is a peptide bond in between the two amino acids. Okay.
Yes, sir. Sure.
So what is the characteristics feature of a peptide bond?
Sir, peptide bond is formed after the hydrolysis of two amino acid. That is the when there is a removal of water molecule, and it always run from N terminal to C terminal. As it is, as we say, it is amino acid that it has both amine group and acid that is carboxylic COOH group. From the COOH, one OH is removed, and from NH2, that is the amino group, one hydrogen is being removed. So when water is removed from that, there is a formation of peptide bond which forms the bond between one amino acid to the successive amino acid nearby.
Rasid, generally, we know a single bond has freedom to rotate at 180 angle, right? Any molecules
Sure,
having a single bond has freedom to rotate by the 180 angle or 360 angle.
sure, sir.
Peptide bond is also single bond, but there is a restrictions in the rotation. That is why we studied about the Ramachandran plot. So how a peptide bond, being a single bond, is not free to rotate all the angles, that is 180 or 360, like other single bond.
So I don't have that much idea, but I can guess it is because of steric hindrance. The uh, the bulkier group we have in the amino acid, it actually propel the the whole length of the polypeptide not to move in a way it can move in 180 degree. Sir.
Okay, is peptide bond truly single bond or some other characters also?
Sir,
Because
simple amide
we
bond.
always represent peptide bond with a single bond.
Yes, sir.
Does it have only single bond character or some other characters also?
Sir, nitrogen has three valency and carbon is a tetravalent. So it will make a share pair of electrons, some things like this, and I'm not sure.
Okay, have you ever studied about the that key peptide bond has a partial double bond character?
Yes, sir, I have heard about it.
Okay,
Partial double
so if
bond.
I ask the double bond character is because of what, then what will be the answer?
So partial double bond because of that unequal sharing of electron between the nitrogen and carbon.
No, this is the wrong answer. The oxygen present on the carbonyl carbon has a lone pair of electron, and that lone pair of electron of the oxygen will involve in the resonance structure. And due to the regenerating structures, nitrogen and carbon, that is peptide bond, has partial double bond character. That's why this single bond peptide bond is not free to rotate about 180 angle. That is why was a concept about the Ramachandran plot, who explain how much angle and how much angle will rotate a peptide bond. There is no freedom to rotate about the 180 angle. Okay,
Okay.
so Ramachandran plot has very significant role and impact for the determination of a rotation of a angle in any protein. So basically,
Okay.
peptide bond, which we generally know that is the single bond, but it is not an actual purely single bond. It has some partial double bond character, just because of the lone pair of electron present on the carbonyl oxygen.
Oxygen.
Okay. Okay.
Sure. Okay, sir.
Uh, for determination of a protein, we have a certain kind of assay. Can you suggest some name of the protein assay?
Uh, Sir, if the protein is the biomolecule under the consideration, then we can go for page, and there are other methods like ELISA. We can go for on the basis of interaction between, if the antibody is a protein, is a type of protein only, then we can study the.
Okay, Rasid, have you heard about the Bradford method or Ninhydin test?
No, sorry, sir. Actually, biochemistry is a nightmare for me. So I'm
Okay.
very. Okay.
Okay. We will move towards microbiology and immunology, right?
Hmm.
Okay. In bacteria, for the gene expressions, we have a that is operon model.
So,
Okay. So how operon model works, and is there a possibility to existence of operon model in eukaryotic cells also?
basically, sir, the best example we have that the lac operon which
Yeah.
we study.
Yeah.
It basically deals
And
with
I'm hoping that
the,
I listen something special about the lag operon to you, because you are
yes,
a
sir.
microbiologist and you have keen interest in the microbiology. So I am
Yes, sir.
expecting that you will answer some unique answers about the lag operon. So
Yes, sir.
you can
I
proceed
will
now.
try, sir. So, basically, lake operon actually a model which helps in understanding the uh, how the bacteria uh, utilizes the lactose as the carbon source.
Okay.
Lactose is a sugar, and beside glucose, lactose there is a disaccharide lactose. How the lactose is being utilized, the carbon source by the bacteria, we can easily study by the use of the lake operon. Lake operon is nothing but it's a model which consists of operator, promoter, and the the Z area that is that responsible for the production of the beta galactosidase, the primary enzyme which is used by the bacteria in order to use the lactose as a carbon source. The-
Okay. So lacoperon is a kind of a inducible type of operon, right?
Yes,
That means it is not always on.
yes, sir.
So what condition make it suitable for on?
Sir, if the bacteria find that in the environment glucose is not in the proper amount, then for the sake of survivability, the bacteria will switch its Lac-Operon model. That is, the promoter will come and it will switch on the Lac-Operon, as a result of which the bacteria helps in the production it get helps in the producing the beta galactosidase enzyme, and this enzyme is responsible for degrading this the lactose, as a result of which it is degraded and it is used as a carbon source.
Okay. For the on and off of lacoperon, which molecule does play crucial role in that switch on and switch off conditions? There will be
So,
definitely there will be a certain molecule which regulate the on and off conditions of that lacoperon.
sure, sir.
So can you name the that molecule?
Sir, is it cyclin AMP, CAMP?
Cyclic AMP.
Yes, sir.
And what about the allolactose and lactose?
Hmm. I'm sorry, sir. I'm not able to recall it right now.
Okay, it's okay, it's fine. Uh, Rasid, suppose there we are trying to stain the bacteria from the examine of gram negative and gram positive, and we follow all the rules whatever we study in our UG and PG classes. But certain suppose if students are doing that and students forget to mix the one step, that is iodine, what will be the consequences in that result? If the students is not able to recall all those things and miss the addition of adding of iodine, then what will be the impact on and consequences of that result? Is will be there any differences?
Sure, sir, it will have. During the process which we adopt in the gram staining,
Yeah.
this the iodine is used as a mordant. Mordant were the prime function of mordant is to intensify the pigment color of the pigment, intensity of the pigment. We add this the iodine after the application of the primary stain, that is crystal violet. If a student he forgot or she forgot to add the iodine after the primary stain, and after just decolorizing it with the alcohol, the whole crystal violet will be washed off by the slide, and only the secondary stain, that is saffronin, which the student is about to apply on the slide, the whole slide under microscope will appear as red. That is mistakenly all the microorganism, the bacteria, that is the gram positive and gram negative, they will appear red under the microscope. Actually, the main role of the iodine is when the crystal violet enters into the cell of the bacteria, it makes the crystal iodine structure with the complex, which as a result of which the primary stain which have entered inside the bacterial cell wall, after the formation of complex, it is not able to come outside the bacterial cell wall. It used to get restricted inside the bacterial cell wall. Those bacteria which have the more content of the peptidoglycan inside, that is, they are gram positive. So if the student is forgotten to add the iodine, the whole slide will appear as red under the microscope.
Okay. Thank you, Rasi. Thank you for a nice interview. And it's your turn. So, if you have any query or any questions regarding your interview, then you are free to ask. So, yeah, अगर आपको लगता है कि कुछ आपके पास सवाल है जो कि आपके मन में है, डाउट है, पूछना है तो आप पूछ सकते हैं।
जी
और जहाँ पे आपको लगा हो कि आपका आंसर सेटिस्फैक्टरी नहीं था, आप
जी जी
जानना चाहते हैं तो उसको आप पूछ सकते हैं। यू आर फ्री।
जी। सर एक्चुअली मैंने प्रिपेयर भी माइक्रोबायोलॉजी और इम्यूनोलॉजी लेकर ही किया था, तो आप आपने बायोकेमिस्ट्री को लेकर ग्रिल किया, तो सर वो मेरा वो मैं वहाँ कन्फ्यूजन इसलिए कि बोर्ड को सर कह देना कि मैंने बहुत लास्ट
मैंने
लास्ट
मैंने
टाइम तक
मैंने
स्ट्रगल
बहुत
किया
सारे
सर।
एरिया से सवाल आपसे पूछे। मैंने प्लांट पूरी पूरी प्लांट बायोलॉजी से क्वेश्चंस पूछा आपसे।
सर
पूरा पूरा सॉयल साइंस से बोलते हैं आपने, सॉयल साइंस से मैंने स्टार्टिंग के 10 क्वेश्चंस लगातार आपसे पीजीपीआर प्लांट और सॉयल को लेकर के पूछा। बट यू
वो
रियली, या बट आपने
वो
बहुत
सर
अच्छे से।
मेरा वो माइक्रोबायोलॉजी से होगा तो सर मैं डील कर लूँगा उसमें। और जैसे बायोकेमिस्ट्री में मूव ऑन करेंगे या मॉलिक्यूलर बायोलॉजी में करेंगे, तो सर वहाँ पे मुझे दिक्कत होने वाली है। तो मैं थोड़ा कन्फ्यूज़ हूँ कि बोर्ड को बता देना कि वो मेरा स्ट्रेंथ नहीं है, तो सर वो
या
एक तरह से
डेफिनेटली
आउट ऑफ।
डेफिनेटली डेफिनेटली आज का एक वीडियो आपने देखा होगा, एक स्टूडेंट है हिमांशु शर्मा,
जी,
है ना? और
जी, जी।
उसने इंटरव्यू जब दिया तो उससे सवाल जेनेटिक्स से पूछा जा रहा था, तो उसने सिंपली ये बोला कि सर आई एम मोर कंफर्टेबल इन इम्यूनोलॉजी, सो इट विल बी बेटर फॉर मी। तो उन्होंने इम्यूनोलॉजी से सवाल पूछा। और
हाँ,
इनिशियली इंटरव्यू विल आस्क टू यू, मतलब ऑफर करेंगे ऐसा ये क्वेश्चंस कि व्हाट इज योर इंटरेस्टेड एरिया, या फिर आपका फेवरेट टॉपिक क्या है, या फिर आप क्या पढ़ कर के आए हैं, क्या प्रिपेयर्ड है आपका। वो ऐसे सवाल को पूछेंगे। उसके पीछे का रीज़न ये है कि उनके पास कोई प्री प्लान्ड क्वेश्चंस नहीं होते हैं। जब आपसे वो इंटरेक्ट होंगे देन डेफिनेटली उस टाइम पे वो सवाल आपसे बना ले, मतलब आपसे पूछते हैं। जैसे सपोज अभी यहाँ पे इंटरव्यू आपका हुआ मौक, मेरे पास कोई सिंगल पेपर नहीं है, कुछ भी नहीं है।
जी,
जैसे जैसे आपसे बात होती गई वैसे वैसे सवाल बनता गया, वैसे वैसे चीज़ें आपसे होती चली गई, राइट? सो सेम थिंग विल हैपन देयर। तो आपको, जब आपको लगता है कि आपका जो कॉन्सेप्ट है वो माइक्रोबायोलॉजी पे ज़्यादा अच्छा है, इम्यूनोलॉजी पे ज़्यादा अच्छा है, तो डेफिनेटली आपको वहाँ पे इम्यूनोलॉजी और माइक्रोबायोलॉजी को ही बोलना है कि सर आई एम कंफर्टेबल इन माइक्रोबायोलॉजी एज वेल एज इम्यूनोलॉजी। वो दो सब्जेक्ट आपके लिए
हाँ,
काफ़ी है। और उनके लिए मैं अगर आपसे यहाँ पे 40 मिनट सवाल पूछ सकता हूँ, तो वो 20 मिनट क्यों नहीं पूछ सकते हैं? आसानी से पूछ सकते हैं
जी,
20 मिनट
जी,
सवाल, है ना? तो आपके लिए ठीक रहेगा। तो अगर आपको
जी सर।
आपसे सवाल पूछा जाएगा तो आप अपने आप को माइक्रोबायोलॉजी एज वेल एज इम्यूनोलॉजी दोनों में बताना आप, ठीक है? और
ठीक है
कुछ?
सर, अगर वो पूछेंगे तो बता दूँगा, और अगर वो मूव ऑन करते हैं जेनेटिक्स और ये सब चीज़ों में, तो मैं उनको ट्राई करूँगा कन्वे करने के लिए कि
बिल्कुल।
सर
आप
मेरी स्ट्रेंथ।
सपोज कि अगर उन्होंने ये क्वेश्चंस ऑफर नहीं किया, डायरेक्टली उन्होंने आपसे क्वेश्चंस पूछना शुरू कर दिया, ठीक है? तो अगर हो सके तो आप उस आंसर को, क्वेश्चंस का आंसर कीजिएगा, लेकिन उनको मूव कर दीजिएगा उधर की तरफ, ठीक है?
सर, सर। जी
तो
सर।
ये
सर
ठीक।
आईपीआर भी मैंने बहुत अच्छे से पढ़ा था।
हम्म।
अभी ये पढ़ा है चीज़ों को बाय सेक्शन। तो अगर वो पूछेंगे कि आईपीआर पे क्या कमांड है, तो सर कह दूँ वहाँ पे कि
Sir,
आईपीआर से पूछे, क्योंकि
to
अगर।
be very honest, for the preparation of a mains, I go through on for the IPR. I have some knowledge about the IPR, but not much confident and not much depth. To be very honest,
ओके,
सर मेरा
ओके,
बैकग्राउंड
ओके।
नहीं रहा है आईपीआर का, तो डेफिनेटली मैंने मेंस एग्जाम क्वालिफाई करने के लिए पढ़ाई किया हुआ है। और
हुँ,
बेसिक्स आइडियाज हैं मेरे पास, बहुत डेप्थ पे नहीं है, बट यू कैन सर आस्क, आई विल ट्राई टू माई
ओके, ओके।
बेस्ट। So such
जी,
kind
जी।
of
मैं
replies
सर।
will also impact positively. That means
ओके।
students have good idea, but still they are trying to
जी,
do something. Okay, so इस तरीके का आंसर जो
जी,
होता है वो काफ़ी अच्छा इम्पैक्ट डालता है इंटरव्यूअर पे। तो आप ऐसे ही आंसर करना। और
ठीक है
डेफिनेटली
सर।
आप आईपीआर के लिए, मतलब आप सीजीपीटीएम की पोस्ट के लिए जा रहे हैं, तो आपसे आईपीआर से एक दो सवाल बनेगा। बट मेजॉरिटी जो फोकस
जी, जी।
होगा वो कहाँ पे होगा? आपके सब्जेक्ट पे होगा। और आपकी माइक्रोबायोलॉजी की समझ अच्छी है, अच्छा लगा, आपने कई सारे ट्रिकी सवाल का आंसर दिया है। हाँ, आपका मॉलिक्यूलर बायोलॉजी का जो पोर्शन है वो थोड़ा वीक है, जहाँ से मुझे समझ में आ रहा है।
जी,
तो आप मॉलिक्यूलर की तरफ आप मत घुसना, आप माइक्रोबायोलॉजी पे ही
जी, जी।
रहिएगा। और माइक्रोबायोलॉजी के साथ साथ में इम्यूनोलॉजी, इम्यूनोलॉजी की भी की कॉन्सेप्ट आपके ठीक हैं, आपका कोई दिक्कत नहीं है,
जी,
आप उन पे आंसर कर ले
जी।
जाएंगे। और जैसे कॉन्फिडेंसली आपने यहाँ पे आंसर किया है, वैसे ही आप वहाँ पे अच्छे तरीके से रिप्रेजेंट करना। बाकी और कोई दिक्कत नहीं
श्योर
होगी,
सर।
मतलब आपका जो आंसर्स देने के टाइम पे जो कॉन्फिडेंस है फेस पे, वो भी ठीक है। और ड्रेसिंग सेंस माशाल्लाह बहुत खूबसूरत लग रहे हैं आप।
थैंक यू सर।
और आपका रमजान का महीना भी चल रहा है, तो कह सकते
जी सर, बेशक।
हैं एक अलग
जी
से
सर, ऊपर वाला करम कर दे
एक,
बस।
या एक अलग से ग्लो है आपके चेहरे पे, है ना? ये बात अलग है कि आपकी बैक की जो लाइट है ना, आपको सही से देखने में दिक्कत कर रही है, शायद डायरेक्शन
जी
थोड़ा
सर।
गलत।
हाँ, ये सर ये है कि आमतौर पे मैं आज ही नहीं हूँ
कोई नहीं,
इंटरव्यू वगैरह
इट्स फाइन,
देने का
इट्स
इसलिए।
फाइन। वहाँ ऐसा कोई दिक्कत नहीं होगा, वहाँ आप दिखेंगे प्रॉपर।
हम्म,
है ना? कब है इंटरव्यू राशिद आपका, 5 को?
सर 5 को है, 11 बजे से।
ओके, तो मतलब
सुबह
आप
7
अभी कहाँ पे हैं? न्यू दिल्ली?
मैं तो सर अलीगढ़ में हूँ।
अलीगढ़? तो मूव कल करेंगे?
कल नहीं करूँगा सर, सुबह जाऊँगा मैं मॉर्निंग प्रेयर के बाद सीधे।
अच्छा, सेम डे
ढाई
मतलब
3 घंटे
फ्राइडे को ही
लगेंगे मुझे।
निकलेंगे?
जी, जी, फ्राइडे को ही
चलिए,
जाऊँगा
बड़ा
सर।
अच्छा है, मतलब ऐसा मैंने सुना है कि फ्राइडे का दिन आप लोगों के रमजान में बड़ा इम्पोर्टेंट और बहुत ही अच्छा दिन माना
जी
जाता है।
सर, इस बार जी सर, रमज़ान के महीने का जो ये आखिरी फ्राइडे जो है अलविदा, बहुत
तो
भीड़ होने वाली है
मतलब
कल हर
सपोज
जगह नमाज़।
आप इमेजिन कीजिए कि आप कितने लकी हो, है ना? कि
जी,
मतलब एक तो पहला कि आपका
जी,
रमजान का महीना चल रहा है, मतलब आप काफी इसको आप मतलब पवित्र मानते हो। और दूसरा
पवित्र
कि वो
मानते
लास्ट
हैं।
आपका मतलब फ्राइडे है ना? तो ये दोनों
फ्राइडे,
चीजें आपके लिए काफी
जी,
अच्छा हो। और गॉड ब्लेस यू,
जी,
भगवान का मतलब
उम्मीद है।
आपके गॉड करें कि आपका बेहतर से
जी,
इंटरव्यू
जी,
हो
जी।
और अच्छे से परफॉर्म करके आएँ
थैंक
आप।
यू सर,
ठीक
थैंक यू
है
फॉर दैट सर।
राशिद, बाकी आपको अपने आपकी कम्युनिकेशन ठीक है, उसमें कोई प्रॉब्लम नहीं है, इंट्रो आपने अच्छा से दिया है। बस आपको थोड़ा बस इसे ही कोशिश करना है कि जो आपकी इम्यूनोलॉजी है, अभी टाइम है आपके पास, एक बार थोड़ा सा फिर से उसको ब्रसअप कर लीजिए, लाइक एज वैक्सीन वैक्सीन के जो टाइप्स हैं उनको कर लीजिए। थोड़ा डिजीज माइक्रोबायोलॉजी को थोड़ा फोकस कर लीजिए, वायरल खास करके वायरल डिजीज पे पकड़ अपनी बना लीजिए। और इसके साथ साथ में इम्यूनोलॉजी में जो आपके थोड़े बेसिक जो कॉन्सेप्ट है, लाइक एज हाइपर सेंसिटिविटी को लेकर के, ये कुछ कुछ टॉपिक है जहाँ से सवाल आपसे बन सकते हैं। माइक्रोबायोलॉजी में जो
श्योर
बेसिक
सर।
चीज़ें हैं वो आपको मालूम है, कुछ स्पेसिफिक अगर आपको लगता है रिसेंटली, तो
जी,
बैक्टीरियल बैक्टीरियल डिजीज़ के बारे में थोड़ी स्टडी कर लीजिए। जैसे हाल ही में अभी
श्योर।
ट्यूबरकुलोसिस डे मनाया गया, तो ट्यूबरकुलोसिस का क्या
जी,
मतलब कैसे हो रहा है, क्या उसके लिए पॉजिटिव पॉजिटिव एजेंट है, ये सारी चीज़ों को अगर आप पढ़ लेंगे तो ज़्यादा बेहतर रहेगा।
श्योर सर, आई विल।
ठीक है? बाकी आप
जी,
इंटरव्यू
जी,
देकर के आइए, तो फिर आप ज़रूर बताइए कि आपका इंटरव्यू कैसा हुआ। और
ज़रूर सर।
हमारी तरफ से आपको बहुत ढेर सारी बेस्ट विशेस कि आपका इंटरव्यू बहुत बहुत अच्छा
जी
हो।
सर।
और ये बहुत अच्छा
जी
कोइंसिडेंस है कि
सर।
आप मतलब ये दोनों चीज़ें मैंने जो बताई, लास्ट फ्राइडे
जी।
एंड एज वेल एज प्लस रमजान का मंथ,
एग्ज़ाम।
तो आपके लिए सब बेहतर हो।
होप सर।
ऐसे ही हम लोग विशेष करेंगे।
जी, जी सर। थैंक यू फॉर द विशेस सर, थैंक यू वेरी मच।
ओके, मोस्ट वेलकम रसीद, एंड गुड नाइट।
गुड नाइट सर।
गुड नाइट, गुड
गुड
नाइट।
नाइट। गुड नाइट।